All right, welcome everyone. Just want to give a brief introduction to Patrick, who's going to be our wonderful host today, telling us more about the evolution and implications of precision medicine with the focus kind of touring through healthcare and anesthesia. So Patrick is a computational neuroscience doctoral student at Boston University and also a super wonderful leadership member here at Simply Neuroscience, where he leads the brain resource creation team. His research focuses on using methods to statistically model neural signals across multiple spatial scales. And he also specializes in frequency domain statistics. He's currently studying how propofol anesthesia disrupts consciousness and also studying the biophysics of Alzheimer's disease under a few wonderful mentors. He is involved in teaching classes and workshops in computational neurobio and biomathematics. English is tough. At the high school and undergraduate level. And some of you may have perhaps seen his different workshops that he's hosted and been disseminating through Twitter. Outside of neuro, he is a regular contributor and enthusiast about the philosophy of technology and educational measurement theory. And he earned his bachelor's in neuroscience and digital and computational studies from Bowdoin College. But is that the correct pronunciation? I always mess that up.
Yep, you nailed it.
Bowdoin, wonderful. All right, without further ado, I will pass the mic over to you.
Awesome. Thanks so much. I really appreciate it. So it's great to see everyone, or sort of see everyone, I guess read everyone's names. So I'm just going to go ahead, fire up my slides here real quick. I'm going to go ahead and share. Is everyone able to see my screen? Awesome. So again, if you have any questions throughout the presentation, definitely just put them in the chat. I'll sort of pause throughout and answer them as I go. I'll also stop at the end to answer questions as well. But I'd love to sort of start this talk off by learning a little bit about everyone who is here. So I have two quick polls for you. So one is, could you share what your current education level is? So in the chat, put a 1, 2, 3, or 4 based off of how you currently identify in your educational journey. And I'll pause for a couple seconds there. Awesome, a lot of different representations. So a lot of high school, some college students. Excellent. Awesome. Well, thanks so much for sharing. And so I'm going to have one more for you. So sort of how comfortable are you with the concept of precision medicine? So 1 being completely new, 4 being I'm an expert and you could be giving this talk, 2 and 3 somewhere in between. Let's go ahead and see. Awesome. So a lot of 1s and 2s, definitely you're in the right place. So today's talk is going to be focused on a pretty high level understanding of precision medicine, going through a couple of examples, one being from my own research in propofol anesthesia. And then we're going to chat a little bit about sort of the implications of precision medicine and its sort of partner term, personalized medicine, and maybe how there are some ethical issues relating to that. But real quick, so you all shared a little bit about yourself. I'm going to share a little bit more about myself since we have a lot of high school students. You're probably thinking about college. So again, I got my undergraduate degree at Bowdoin College, which is a small liberal arts school in Maine. It's about 3 miles from the shore. And while I studied neuroscience and digital and computational studies, which I sort of united in my PhD studies, I was able to take a whole bunch of different classes, which was really rewarding and sort of I credit a lot of my current research to now. So I was able to take classes in government and legal studies, in sociology and philosophy and education. And so I think a lot of those things really made me a much better scientist. And so I definitely just want to quickly plug that you don't have to be a STEM major or even a neuroscience major to do neuroscience research or neuroscience related research. And we'll sort of touch on that in a little bit. But again, I'm a PhD student, so I'm in my second year doing computational neuro at BU. And I have quite a few research interests, as we mentioned earlier, but the one that's most relevant for today is I study propofol anesthesia and specifically how it disrupts consciousness in both humans and macaque monkeys. So monkeys are a great animal model for understanding a lot of similar cognitive processes, or at least the proxies for behaviors in humans. So I'm sort of going to be leaning on that for a lot of my talk towards the middle. One question that I do want to address here, and it's relevant to precision medicine, is what is computational neuroscience? And so I've gotten a lot of emails throughout the week, and so I thought I would just take some time in my talk today to actually explicitly address it. So folks were sort of suggesting that it might require particular skills or particular coursework to be able to sort of qualify or fall into this category. But that's actually not true. So computational neuroscience is more of like a vibe in how you approach your research. And it typically requires two things. So one, you're just going to sort of abstract particular features that you're interested in from real biological systems. And really all that means is you're just going to pick what you want to highlight. So for example, if you're interested in a single cell, right, they have ion channels of different types. You might only pick a few of them to focus on. And by explicitly focusing on those things, you ignore everything else. And so it's important to sort of understand what you're actually including and what you're leaving out when we're sort of interpreting our data. But what makes it computational is whatever we're abstracting, we're doing something either mathematical or computational with that data or those models. So it could be something based on equations. So for example, differential equations, that's a traditional way of sort of approaching data. But we also could do something more contemporary with machine learning algorithms, or something that's been around for a while, deterministic algorithms, to sort of gain insights about those features. And so real quick, mathematical biology goes all the way back. You might have an action potential. We might be interested in 3 cells. So for example, or cells, ion channels. So for example, a leak channel, calcium channel, and a potassium channel. We're making explicit decisions about what we're including. We'll write equations to describe those, and then we're able to sort of model action potentials from a cell. Similarly, we can do this at larger scales, which is something we're going to be talking about today. We can collect really high dimensional data, apply different algorithms to it to uncover insights into the data, and then sort of make claims from there. Same thing with bioinformatics, which is in the field of genomics, where we can collect a lot of, for example, RNA sequence data, apply different algorithms to that, and make claims about if a particular gene is, for example, upregulated or downregulated relative to some reference. So all that is to say is computational neuro is more of a way of approaching problems rather than sort of a specific set of skills you have to be able to do that research. And so that sort of brings us to what we're really interested in today, which is this idea of precision medicine. And I think the best way to sort of get into the topic is by going through a brief history, albeit a crude history of medicine, to sort of understand where we have been and where we're going in the future. So I'm going to focus mostly on the 20th century, where towards the start of the 20th century, we typically did something called intuition medicine. And so this is where doctors would sort of get extensive training in the clinic where they focused on identifying particular signs and symptoms as they relate to an underlying disease. And the medicine part of it focused on trying to address those particular symptoms. And so, for example, if we think of COVID-19, there are many different symptoms that are associated with it. So it might be a high fever, it might be sore throat or congestion. There are neurological symptoms with it. So for example, you might lose your sense of taste for a while, or sense of smell. On your skin, you might develop rashes, for example, on your toes. So these are all symptoms that are related to a particular underlying disease. And so what we typically did is we tried to address those particular symptoms without actually getting, in every case, at the underlying disease. And as you can imagine, an issue with this approach is that even though the underlying mechanism might be the same, symptoms manifest very differently in a lot of different patients. So some patients might not have any of those symptoms. They might have had unique symptoms like nausea, or you might have had all of them. And so it was sort of like a piecemeal approach that relied on the expertise of whatever doctor you had. And so the quality of care you received often relied on that particular individual or the care team that was available. So around the late 40s, early 50s, we started approaching medicine in an evidence-based way. And all that is to say is we started using clinical trials. So we started randomly assigning individuals to treatment groups to test and gain information about a particular drug. And we compared that to control conditions where individuals didn't receive a particular drug. And we tried to make claims in the aggregate about how safe and effective a particular drug was. And even then, even though we were focused on sort of the quality of the treatment, we also just made a very crude list of every sort of health outcome that could occur. So whether it was from reactions to the drug, or if someone passed away for other causes, but they were on a particular drug, it would still be sort of included in our description of the symptomologies of the drug. So we sort of just took a very conservative approach, tried to describe things in a lot of people, and then if in the aggregate it was safe and effective, we would sort of proceed with those sorts of treatments. Now you can imagine the problem with that is even though we're getting a lot of data on a lot of different people, it's not sort of tailored toward a particular individual. And so even though a particular drug might be effective for many different people, there could be large subsections or cross sections of the population that either had severe reactions to the drugs or didn't get any added benefit, even though they were taking on the risk. And so this is basically how most contemporary medicine is still done. And so if we continue with the COVID-19 metaphor, we can think of this either in preventative care or actual treatments. So preventative care for COVID-19, you might think of vaccines. So we know that they're very safe and effective in preventing hospitalization and progression of severe disease. But that wasn't the case for every particular individual, especially for people who tend to not respond well to vaccines. And in terms of treatment, we have great antivirals for things like COVID. So for example, Paxlovid is highly effective for a lot of people, but there are plenty of people who do not receive any benefit from it and in fact get a lot of negative side effects for the drug. And so this sort of leads us to think about, what else could we do? Is there a way to get a more tailored approach? And so that's where precision medicine comes in. And so there's a lot of different definitions of precision medicine. We're going to sort of give a general one here and then sort of nail down a more precise definition for it. But more broadly, precision medicine sort of accounts for an individual's variability in genes, lifestyle, and environment. And why that's relevant is we're interested in sort of how those things will sort of lead to a disease manifesting differently relative to some of their peers who don't have those particular clinical profiles. And so through this definition, we might think that precision medicine is interchangeable with personalized medicine, but it turns out that that's actually not true. And we're going to get into that in a little bit. But you might have guessed correctly, and we'll unpack this in a little bit, that precision medicine requires a lot of information about not only an individual, but many, many different people. So we can understand how particular features of those people interact with, a treatment approach. And so it's not just to say that, XYZ drug is effective for this particular individual. We need to abstract certain features, such as someone's genetics or their lifestyle choices, and understand how that actually impacts the progression of a disease and how effective a given treatment is. Now, before I go further, I do want to spend one more second on, why not evidence-based medicine? Because you might sort of get from the last figure that we need to sort of move beyond it, or that it's obsolete, which is also not true. We're basically trying to build off of our historical approaches of evidence-based medicine. But what we're actually changing, instead of just focusing on an individual person entirely, we're trying to get better cross sections of the population. So in evidence-based clinical trials, we'll sort of get many, many different people from diverse backgrounds, and we'll try to make claims about the average person. But again, remember that average person doesn't exist. But in precision medicine, we're going to tailor it more towards smaller subsections of the population, where one treatment approach or drug might be more effective than another one. So we're just sort of improving on this approach rather than throwing it out. Now, when we dig a little bit further, we'll see that precision medicine can actually be distinguished from something called personalized medicine, even though in sort of how we normally talk, they might seem interchangeable. And the difference is actually quite profound in a couple of orders of magnitude in terms of the data that's required. So precision medicine is more of a tailored treatment approach, whereas personalized medicine is more of a sort of custom treatment. And so what I mean by tailored is what I was sort of just alluding to on the last slide, where we're interested in particular scales of an individual. So we might be interested in their genetic profile, their molecular or cellular profile. If we're talking about, for example, cancer, we might be interested in the histology of a given tumor, or anatomical differences at the individual level. And so using these scales, we can sort of group people together better than using just crude metrics such as age, biological sex or gender expression, or race. So we're not sort of really focusing on any particular individual, but just smaller shared subsections of the population. Personalized medicine is what most people probably think they're talking about when they say precision medicine. And it's sort of that, like I mentioned, the custom treatment. So this accounts for everything in precision medicine, but it also uses environmental, socioeconomic, and psychological experiences in addition to those the biological dimension to understand sort of how a particular disease can be treated, and how effective particular treatment approaches can be. And so I want to give two examples in a second before we move into our case study, but I just want to underscore this point because it's going to be helpful in our ethical discussion, where precision medicine and personalized medicine require making sense of far more data than we ever have before. And so as you'll guess, we're going to talk a little bit about data treatment, who owns the data, what happens when people don't want to give up that data, should we even collect that data, and things along those lines. But it's just worth saying pretty early on. So even though precision medicine is going to require all that data, it seems like it's a tall task. We need to include many, many people in sort of these clinical trials. I do just want to point out that we already do it in many forms of medicine, where sort of the most successful approach would be in oncology. And so when we're trying to treat many different types of cancers, we've sort of moved away from just treating symptoms, or B, just sort of taking the most aggressive approach we possibly can. We sort of take a great deal of time to understand someone's particular manifestation of cancer, and sort of take a tiered approach to treating it. And so in some cases, right, if we do the work to understand what type of cancer someone has, in some cases, just surgery is required to remove it, and then we'll sort of monitor from there. And so this is great because you might be able to treat a particular form of cancer without sort of taking all the extra risk of all of the other treatment approaches. Whereas in some cases, people will have more aggressive forms, more widespread forms, and we're able to take more aggressive approaches. And so this idea of precision medicine in oncology and other related fields is basically tuning the risk to benefit ratio for how we are approaching a particular individual, based off of the genetic profile of the cancer that they have. And so that's sort of a hand-wavy way of discussing that. But it's basically, we understand how certain forms of cancer based off of where they are and their genetic profiles will progress, and we can sort of pick the best treatment schedule from there based on that. Now, this doesn't account for a lot of different diseases, right? So precision medicine is limited in some regards where we're hyper fixating on the actual individual and their individual case of cancer, and we're sort of ignoring the sort of things that might contribute to their disease progression outside. And so an example that isn't related to cancer, but rather heart disease is someone's environment. And so one pretty striking fact is that environmental noise, for example, in Europe, can contribute to 48,000 new cases of heart disease each year. And there's similar figures in the United States as well. And that's sort of because it will interact with underlying conditions in an individual. So for example, like hypertension or genetic factors that predispose someone for heart disease. But even though people have those particular factors, it's the literal environment that is contributing greatly to that disease progression. And so in this case, we wouldn't sort of get a sufficient result with precision medicine, but rather we need to focus on more structural or policy-based changes to improve health outcomes. So hopefully that sort of gives us an idea of the distinction there. And so that also leads to this other discussion, which I was sort of alluding to at the start of the talk, where it's not just sort of medical professionals who are interested in doing precision medicine or personalized medicine research, but rather it's sort of a whole swath of professionals who contribute in their own unique ways. So we have folks like myself, where we have basic science research that's being done to understand systems, understanding how particular genes are implicated in a given disease, understanding how particular biomarkers relate to the progression of a disease, and so forth. But we also have folks like social scientists, where psychologists might be interested in how mental health impacts someone's disease progression or their ability or interest in receiving care, or even their ability to give informed consent in receiving care. Whereas sociologists might focusing on more structural issues that might interact with someone's health outcomes. So for example, structural racism in the US is a really important topic right now, but it also could be something such as, where are houses located in relation to public transportation? Obviously, as I mentioned, we need a lot of different data. So we need many people who are able to sort of pull out interesting insights from the data that we collect and sort of communicate those to stakeholders. Obviously, we have our medical professionals, but then also philosophers who are interacting with many different sort of types of ethical questions, which we'll touch on in the end. But as I take a rest real quick to grab a drink of water, just so I can get another read on the room, could you put in the chat sort of what you're interested in out of those areas? You can put more than one, but just put a 1, 2, 3, 4, or 5, just so I can tailor the discussion at the end. Awesome. Yeah, this is such a great sort of cross-section of folks. So a lot of healthcare professionals, some natural sciences, some people who are natural sciences, computationalists, and so forth. Yeah, great. Thanks so much for sharing. Cool. So I do want to take the time, as I mentioned, to sort of go through a case study of propofol anesthesia and how we can improve anesthetic-based care through precision medicine. And I have up on the slide here in defense of precision medicine, because it might seem that the logical choice, given the choice between precision medicine and personalized medicine, would be to do the one that's more customized. So the one that goes to the smallest common denominator. So whether that's, actually individualized care or very, very, very small cross sections of the population. But I hope to sort of convince you that precision medicine in many cases might be sufficient for what we're trying to get at. Real quick about propofol anesthesia. So it's a general anesthetic. It's widely used. It's one of the most commonly used anesthetics. So if you get, for example, relatively invasive surgery, you've probably received it. In some cases, less so now than it used to be. But if your parents or you have ever gotten like a endoscopy or colonoscopy, you might have been under propofol anesthesia. And so you're fully unconscious when you're at higher doses of the drug. And it has a lot of nice properties where very few people are allergic to it. And when you cease your infusion, it's going to sort of decay on its own and people will return back to a sense of awareness. And so it has a lot of nice properties. We know quite a great deal about what it's doing at a molecular level. So it's a GABAergic anesthetic. So what that means is propofol is going to bind to a GABA A receptor, which the long and short of it is chloride is going to rush into the cell, which hyperpolarizes the cell. And so for folks who are familiar with those words I just threw out, it basically means that most cells that are affected by propofol are going to fire less regularly. And so in a sort of global sense, we think of anesthesia as sort of quieting network dynamics. One way that we study this, at least in sort of a monitoring sense, is through electroencephalography or EEG. It's something or a tool we've had for a very long time. So it was discovered in 1929. And basically it's a direct measure of electrical activity in the cortex of the brain. And specifically what we're recording from is, again, from the cortex, these pyramidal cells. So on the picture on the right here, you see a bunch of different neurons that are all sort of lined in the same direction. They look quite beautiful, in my opinion. But we're lucky that they're all oriented that way, or most of them are oriented that way, because it creates something called an electrical dipole, which is actually what allows us to record meaningful brain activity from the cortex. Now, EEG only records from the cortex, but that's okay, because in propofol research, we're interested in a lot of behaviors that are driven in the cortex, primarily in the cortex, and then obviously it interacts with subcortical regions like the thalamus and things along those lines. But we can record meaningful activity from the cortex non-invasively. So typically EEG will just be a cap that people can wear if you have a lot of channels in an anesthetic context, specifically in clinic. You'll typically just put like a sticker on the front of the forehead, and you're able to have a couple of channels that will record EEG from frontal cortex. But it's relatively inexpensive, it's non-invasive, it's comfortable for people to be recorded from it. But it gives us, again, these measurements from these electrical dipoles, and that results from sort of the current flow within neurons. And so if you have questions, again, about the electrophysiology underlying this, definitely just reach out. I'm happy to chat about it more. But what's really important is that we're able to get a lot of really good high-dimensional data that comes from many, many, many different neurons from the cortex. And so why that's relevant is in anesthesia, right, we're sort of globally affecting the cortex. And so we're trying to get information for what the cortex is doing, what these networks are doing at a very sort of gross or high-level scale. And so why this is exciting is using EEG under propofol anesthesia, we're able to potentially detect distinct states of unconsciousness through what are called spectral biomarkers. And so I'm not going to unpack this entire figure, but what's really important is that as we sort of progress through different doses of propofol anesthesia, so from not receiving any to a low level, which is considered typically sedation, low dose to high dose, we're able to do two things. One, record our EEG, but then also do a behavioral test. And so in this particular study, participants were asked to respond to two different types of stimuli. A particular sound which would cue them to respond to or respond with a word. So for example, they would hear a beep and they needed to respond back verbally. And then when they heard a different sound, they were asked to sort of click a button. And so we see that those two behaviors can be decoupled from each other. But in this particular study, when both of them sort of stopped occurring, so people were not responding, that would be defined as the loss of consciousness. And when they started doing both of those things again, that was the return of consciousness. So that's a crude way of sort of understanding these initial states. But what was really exciting is by doing something called phase amplitude coupling with the EEG data, we were able to parse apart clinically two different stages of unconsciousness. So we were able to see with the start of this lower dose, we saw this phase amplitude coupling. And for our purposes today, we can just sort of look at the color scale. So it's very red here in more posterior regions, but nothing occurring in the more frontal regions. But as we proceed to higher doses, the phase amplitude coupling occurs both in the frontal and posterior regions. And so what's really exciting is what we're interested in good anesthetic care is avoiding these really higher doses where people will go into something called burst suppression, which is basically just shutting down as much activity in the brain as possible, which can lead to a lot of negative outcomes. So severe nausea is probably the most common one. But we want to avoid that. But we also want to avoid being at such a low dose where people are going to wake up when they're in the middle of surgery. So through these spectral biomarkers, we're able to understand in a particular individual what is appropriate dose to keep them in between those two levels. And we're getting even better. So this is a paper from, I think it was 3 or 4 days ago from one of my colleagues, where just with only their frontal sensors, they were able to detect a different type of spectral biomarker, where they see this greater intensity in the alpha band and low beta. So those are just predefined historical frequency ranges. And we see as propofol increased, the red band sort of tightens up, gets more narrow, the colors get more faint. And then we get this period where there's almost no red in that particular frequency range. That's the burst suppression we were just talking about. And then it sort of ramps up again as the dose decreases again. And so we're able to get a pretty good idea of what the cortex is doing when people are at different doses of propofol. And so the application of this is we can sort of hook it up to a computer, and in a closed loop fashion, we can make sure that people are getting the right amount of dose when they need it at a 20-second interval. So we can sort of check in almost a real-time sense to understand, is someone getting enough anesthetic to keep them in an appropriate level of unconsciousness. And so this is where the precision medicine part comes in. We might start with some initial target level of propofol. So this is the moment of unconsciousness, MOU. So we might start with some initial dose. And then what we're going to do is we're going to look for that biomarker with the EEG that we just looked at. And when it's detected by an algorithm, it's going to try to infuse the right amount of the drug to keep them at that particular level. And so in this example, we're doing it on non-human primates. So one of my colleagues was testing this in animals to make sure it was safe and effective. But we're able to, in a closed loop way, in an automated way, make sure that people are getting the care that they need based off of what we're physically seeing in the individual. And so this is a case of precision medicine because we're looking at a particular biomarker in an individual, but it's not personalized medicine because we're not interested in the other areas of their life. So for example, the environment that they live in, their lifestyle choices and things along those lines, because we can just sort of track it by looking at this biomarker real time. And so this is a case where it's sufficient to just deal with precision medicine as opposed to personalized medicine. But what's important with these sorts of approaches is it's one thing to identify brain states, but we also need to understand what they actually mean. And so what I was chatting about, we sort of are able to make those inferences based off of what people are doing behaviorally and what we know is occurring in terms of the actual dose of propofol. But what researchers like myself do is we try to understand, well, what's actually happening across layers of the cortex, what's happening in relation to thalamus and things along those lines. So we actually have a really good idea of what those spectral biomarkers mean biologically in a person when they're under anesthesia. So it's one thing to sort of get these biomarkers, it's another thing to actually know what they mean. So some takeaways so far. Precision medicine is sort of a highly reductionist approach to health because we're just focusing on sort of these biologically relevant features in a person, but we might not look at their greater context, so their environment, socioeconomic status, and things along those lines. Being reductionist isn't always bad. So in some cases, it's sufficient. And why we would go for sufficiency over, for example, incorporating everything to just see is that collecting a lot of data and trying to analyze it to get meaningful insights is one, it's difficult, two, it's very expensive, and three, it's very invasive to individuals' lives. So we'll get into that in just a second. But for most diseases and procedures, precision medicine is too simplistic. So even though today's talk is all about precision medicine, I sort of wanted to take the opportunity today to draw the distinction between the two of them and point to the fact that precision medicine isn't going to solve all of our problems. We need to understand sort of the structural forces around a particular individual to provide appropriate care. So other considerations, so this is sort of getting at there we go. So the ethical dimensions that we were talking about. So there are countless considerations that need to be addressed before precision medicine sort of develops further and is found in typical care that people receive. And I've sort of just done a brute force list here, but I'd love to sort of go through each one. And if anyone has any questions or comments about them, definitely put them in the chat. But a big one is the misinterpretation of data. And so even though we live in an era of big data where we're sort of collecting everything we possibly can get our hands on, we're making it more publicly available than ever. We have a lot of computational tools that can try to analyze them. More isn't always better. And so in a lot of cases, right, we might have a lot of different things that are easy to measure, but might not tell us anything clinically interesting about someone's care. So what we need to do is be very careful in sifting through all the data that we have to make sure that it's actually relating to the outcomes that we're shooting for. So that's particularly relevant in today's day and age with, for example, machine learning algorithms, where they're excellent at figuring out patterns in the data, but they're terrible in the sense that the parameters that help fit those models and give us the results aren't always readily interpretable because it uses as many parameters oftentimes as necessary to create or sort something into a particular category that we're interested in. And so that's why a lot of those older methods, so for example, using differential equations, are really helpful in certain settings because we are in full control over the sort of assumptions that we're making about the system. And so even though it's not capturing everything as well as machine learning, the conclusions we make are explainable. And so that's something that precision medicine is going to need to figure out because we have a lot of data that individuals can't sort through in a systematic way very easily. So we need to solve this interpretation problem. Another problem is in terms of patient stratification. So obviously the whole point, as we talked about throughout today, is that precision medicine and to an even greater extent, personalized medicine will take our traditional evidence-based approach and then draw finer and finer squares around subsections of the population to identify relevant features that group them together and then identify the best possible treatment route for a given disease for those folks. A lot of diseases are rare in the sense that they don't happen at the order of hundreds of thousands or millions of people. but it could be, a couple of thousands. And so even though we get a lot of headlines for the more prevalent diseases, there are a lot of folks doing research in those areas. So it's possible that precision medicine and personalized medicine aren't possible in those cases because we aren't able to get enough information from enough people to cover all of the bases that we might be interested in to improve care. So in some cases, evidence-based medicine might be as good as we can do if there aren't, a very sort of, if there isn't a more diverse sort of population with that particular condition. Another thing that we really need to think about more on a policy standpoint is the potential misuse of data. And so some folks who might be really interested in all the data required to do precision medicine are insurance companies and for-profit health technology companies. And so, in a lot of contexts, these folks do a lot of important work, but also they do have a history of taking particular sets of data and making decisions that might not be in the best interest of patients. So, for example, insurance companies oftentimes will collect data from the clinic where patients are asked, have you fallen in X period of time? So like the last 30 days or something along those lines. And if you have, it's an excellent predictor of whether or not someone is going to be a fall risk in the future, particularly in 2 to 3 years. But it also often leads to higher rates for individuals. And so if we tie this into, for example, misinterpretation of data, insurance companies or for-profit companies or even nonprofit companies might take information that might not be very meaningful, but might jump the gun and make decisions about someone's, for example, insurance policy or something along those lines that might not actually reflect reality. So it's important to make sure that everyone who's involved in the process is responsibly using data. Another thing, and this ties into sort of the psychological side of precision medicine, but we need to make sure that folks are giving informed consent. And so this is not just in the sense that people need to be aware and be able to okay something, but they need to sort of understand the full repercussions of what precision medicine and personalized medicine entail. And that would be the collection of large swaths of data that might be able to identify them. So if someone has a very rare illness, and they have particular features about them, even if the data is sort of decoupled from their literal identity, oftentimes it's relatively easy to figure out who is behind the information on a spreadsheet, especially in today's day and age. So we need to understand, or we need to be able to communicate precision medicine to the patients to make sure that they understand what they're getting into. And then there's more tangible aspects. So data management, right? We have all this data. We need to understand how do we not only store it effectively, but also make it usable. So the databases need to be able to interact with the algorithms we create and the platforms that we're interested in using to make sure that we're actually able to extract the insights that we're hoping for, rather than having a bunch of different platforms that might be incompatible with one another. And we also need to make sure that we're securing patient information in a way that I think sometimes is swept under the rug a little bit. So where we're actually going to store all that data is really important. Finally, access goes without saying, right? We already have access issues in many different countries where folks of particular demographics, so whether it's low socioeconomic status, or particular racial categories, might not have the same access and quality of care as some of their peers. And so we want to make sure that everyone who wants to be represented within a particular data set and treatment strategy is represented. So we need to make sure that we sort of have an inclusive lens right from the start. But so that was sort of my very quick crash course of precision medicine. Towards the end here, I do want to sort of touch on a couple of skills that might be relevant for folks who are interested in doing precision medicine research or personalized medicine research. So for the high schoolers in the room, it's really important to develop these skills whenever you can. You don't have to. They're not prerequisites for doing this work, but I find that a lot of people in my field do really well with learning coding early on, just because sometimes it can be a little intimidating. So starting slow and accessibly would be a great way to approach it. 2 great languages for free that you can start just like on your own computer, or even in some cases the phone if you're watching on a phone, are R and Python. A lot of great resources online for that. If your high school offers mathematics courses in statistics and calculus, definitely take them. If they don't, try to take like the highest level of math you can. That is always really helpful, and it'll get you a good start on your sort of college education. In terms of scientific writing, you can start a journal club, not necessarily reading through crazy scientific literature or anything along those lines, but finding credible blogs that you're really interested in, or documentaries that you're interested in, and trying to sort of go through them with peers, talk about findings, relate them to society and things along those lines. If you aren't able to do that, getting involved with writing-based activities at your school can be really helpful. So, a student newspaper, or if you want to start a blog, if you don't have those resources at your school, you're able to do that. And finally, communication is really important. So I think at this point, it's more of learning how to ask for help from teachers, from community members, from mentors, things along those lines. Understanding your needs and communicating them to people who might be able to help is something that is really important in the field, especially in my case, right? I only know what I know, and my field is full of experts from many different areas that we all have to sort of collaborate to be successful. And so the sooner you're able to work with people, communicate what's missing from what you're trying to do and how they can help, the better. So in terms of college students, it's sort of the same overall theme. So the same languages, things along those lines. But you might work on, simulating data. So for example, EEG data in the case of propofol anesthesia, or fMRI data. You could also do more biophysical processes and things along those lines. Bioinformatics, as we talked about earlier, there's a lot of great tutorials for that. But sort of understanding these practical problems and how to use these languages to answer them, that would be great. In mathematics, if you can take linear algebra, that's far more important. If you can only take linear algebra versus calculus, I would take linear algebra. Signal processing is really important. That's pretty much most of what you do with these time series, so definitely get your hands on that sort of things. And sort of continuing this thread of scientific writing, communication, and things along those lines. You really want to sort of go up to the next level, where you might be able to, say, volunteer in a lab, or work in a lab in a paid position to either write a manuscript or at least practice writing a manuscript. That would be really great. There's a typo in terms of starting a journal club, but start or join a journal club. Again, those are more frequent at undergraduate institutions than, for example, high schools, but definitely recommend doing that. And communication, this will be helpful not only in your scientific writing, but also just classes in life in general, understanding sort of like the different levels of difficulty at which you need to, well, how do we say this? This is so ironic that I'm messing up here. So the level of difficulty or level of detail that's required to communicate to the audience that is in front of you. And so, for example, today I was very sort of hand wavy, try to draw on various like high level examples, but that's going to be very different from how I talk to my colleagues or the medical doctors that I collaborate with to actually further our research. And so knowing how to approach those situations is going to be very important. And then finally, working in a lab. I just listed 3 examples, but literally whatever lab you can get into is a great way to sort of dive into research and sort of figure out what you're interested in. So I really appreciate everyone coming today and sticking through this. I apologize if I'm losing my voice a little bit. I just am getting over a little bit of a sickness. But let me know if you have questions in the chat. You're welcome to unmute if you have questions. But if you think of anything down the road, you can reach out to me on Twitter. or over email. So you can just send me a note and I'll respond as quickly as possible. But other than that, thanks so much for joining me.
Thanks, Patrick. That was a really awesome and detailed overview. Learned a lot. Like Patrick mentioned, folks, if you have any questions, feel free to drop them in the chat. Or you can also raise a hand and unmute, whatever you prefer. Maybe everyone is starstruck and has no questions, and that's okay too.
Yeah, thanks for coming out, everyone.
Now, where can I find resources to learn R?
Yeah, definitely. So, Jessica, and I'll put my email in the chat again, definitely send me an email and I'll just send out all of them that I have. So there's going to be a lot of options. So some of them are just like nice online courses with some lecture videos. Those can be helpful. Some of them are going to be neuroscience specific. So there's a lot of really great resources that have come out in terms of like basic neuroscience simulations, data analysis. And what's great about them is they always start from you assuming that you don't have any knowledge. So definitely anyone who's interested in R or Python or even MATLAB resources, I'll just send you like a whole a list of resources. I'll pop my email down here. Yeah, and in terms of the not responding, don't worry, I am going to respond. I have time set aside tomorrow. So this week, I have a National Institute of Health grant due pretty soon. So I've just been grant writing all week, and so I've been kind of trying to chug through that. But now that I have a full draft of it, which I'm very excited about, I'm gonna be able to comb through those emails. So definitely keep an eye out tomorrow. Yeah, great question. So the question is, so how does R differ from SPSS? So SPSS is a form of statistical software that's like controlled by a company. So they have, for example, a like graphical interface. So a GUI is the acronym for it. But they basically have a limited number of statistical tests that you can do on particular data types. And what's great about it is that because someone is managing the software, you don't have to sort of code those things yourself. You don't have to write those equations yourself, like in code or anything along those lines. You have a lot of flexibility, powerful data visualization techniques and things along those lines. So traditionally, folks in psychology will typically be taught that. And I think at a lot of universities, even to this day, like students are able to get certified in SPSS. So it's a great way to sort of understand data analysis. A lot of professionals still use SPSS. But programming languages like R and Python are more general in that you don't have to use them for statistics. They can be used to make like websites and things along those lines. So they're very flexible. But those languages can be used for either creating custom analyses that might not sort of exist in SPSS, or they can also be integrated into automatic pipelines that can sort of allow you to do very large data sets, complex analyses and things along those lines without you having to interact with the software. So you can sort of write the Python pipeline, just let it run and kind of go from there. So I think that would probably be my high level distinction between the two. They're both great. And so if you only have an option to learn one or the other, or if you can only learn SPSS, that's fine. If you have the option to learn one or the other, I would learn something that's more general, so R or Python, just to give you more professional flexibility inside of neuro, but also outside of science in general. Yeah, so this is in response to Jessica's question. So what are your preferred APIs? So I'm assuming you're talking about like IDEs, so the environment that the language runs in. So for R, I just use RStudio, and for Python, I use VS Code, which is from Microsoft, and it's a really powerful. It can be difficult to get set up with initially, but there are a lot of great resources online for starting those up. If VS Code is really disorienting, which it was to me, Spider is a really great intro IDE. So there's just a couple of panels. You don't have to do a lot of configurations. Basically, once you install that in Python, you can just jump right into it. But in any case, I would definitely have you go through, for example, YouTube tutorials. Typically, you can get started in like less than like 20 minutes, but it'll be helpful for avoiding any traditional startup errors. So once you get it installed and set up, it's you're ready to run with it. In terms of are there any sites that provide raw data that we can work with and manipulate? Absolutely. So there's a lot of free data online. Historically, GitHub, which is sort of normally used for like software development or like managing different versions of your code with a team. You can also post data files on there. And so UCSD, so the University of California, San Diego, has a huge data set of EEG data in different disease states. So for example, like Alzheimer's patients versus control subject or participants. There's some with schizophrenia versus controls. Similarly, I think UC Davis has a lot of bioinformatics data. So if you do some Googling and just say like GitHub next to the type of data that you're interested in, I'm sure you'll be able to find some great free resources, but they are out there. I will warn you that it might be difficult to understand how their actual data is structured. Sometimes there are directions on how to deal with that. Sometimes there aren't. But if you run into any trouble in finding something that you want to play with, just like reach out to me, and I'm happy to help you find something that works for you. Cool. I think questions have sort of slowed down. So again, send me an email if you have anything else, but otherwise, I think maybe we can wrap it up.
Good. Thanks, Patrick. And thanks everyone again for joining. I know some of y'all are up really early and up very late as well. So I really appreciate you guys tuning in live. And we'll be wrapping up tomorrow. We have, I believe, 2 more presentations, one in the morning by Edwin and one in the afternoon by Pooja and Shruti about finding collaborators and mentors. So hopefully we'll see you all in the morning or the evening, depending on where you are. Take care. Bye, everyone.
Thanks, everyone.